So a number of you had commented in your weekly reflections from this week that the idea or the concept of a degenerate primer was the most confusing from this lecture. Not hard to figure out why that was. It's the last slide of the day. We were running a bit short on time. Very much went through that far quicker than it should have gone through. And whenever something comes up so uniformly and there's so much of a consensus over the complex topic, something that was confusing, it makes sense to review it again. Only problem here is I don't want to review this in class because I don't want to detract from sequencing this week, so I figured I'd just throw together a quick video and cover it that way instead. So in the next few minutes here I'm going to try to explain this a little bit more clearly and a little bit more methodically than I was able to do in class. And let's start with what we mean by a degenerate primer. So remember, we talked about the number one limitation of PCR being the need to know the sequence ahead of time. In order to conduct a PCR, you have to have primers. In order to design primers, you need to know the target sequence that you're going to be amplifying, and that means that you need the sequence of your target DNA prior to PCR. The one real workaround to that is even remotely elegant is this idea of a degenerate primer. So when you know the protein sequence, that is the amino acid sequence of your protein, of course you know that there's a gene in the genome somewhere that encodes that protein, otherwise it wouldn't be synthesized in the first place. You know that that gene exists, but you don't know the gene sequence. You just know the protein sequence. You just know the amino acids. So the idea of a degenerate primer is designing a pool or a collection of heterogeneous primers where within that pool you are absolutely certain that one primer will anneal to the gene for this protein. You just don't know which primer that is. In other words, you cover all of your bases by designing a large collection of primers where you know that many of those primers won't be correct, won't be complementary to your gene of interest, but one of those primers will be able to bind, will be able to anneal. So we're starting this idea here with this very, very short protein sequence. The amino acid histidine followed by phenylalanine, proline, another phenylalanine, methionine, and lysine. And these 6 amino acids make up the beginning of the protein, let's say, and we're going to design a primer to anneal to the DNA sequence that codes for those 6 amino acids. Now let me say that again. We're going to design a primer that has a DNA sequence that will correspond to that protein sequence of 6 amino acids. The problem again is that we don't know which specific sequence we should design, so we'll design a set of heterogeneous primers where one of the primers will definitely bind to our gene of interest. To do this properly, we need the genetic code. The genetic code is there provided on the slide. And we go into this knowing that if histidine is the first amino acid of the protein, the first amino acid we're targeting with our primer at least, we don't know exactly what codon is in our target gene for histidine, because again, we don't know the sequence. But we do know that histidine is encoded by one of 2 different codons, either C, A, U, or T in this case, or C, A, C. So we're going to design our primer to capture both of those. We're going to design a primer, in other words, that has C, A, T in one position, or C, A, C. Now that's the degenerate primer. Let me back up for a second here and just point something out. When I say we design a single primer, that's a misnomer. It's a misstatement in a way. We're actually designing 2 primers. Remember, it's a heterogeneous mix of many primers. And one of the primers in our mix will start CAT, and the other primer will start CAC. We know, since those are the only 2 codons for histidine, we know that one of those 2 primers will anneal to the first 3 nucleotides of our gene, because those are the only 2 codons that encode for histidine. We just don't know which one of these will be the one that works. And then we go back to our sequence, and we see that the next amino acid is phenylalanine. Well, phenylalanine can be encoded by, and I'm going to stop saying U's because we're talking about the DNA context, so it can be encoded by TTT or TTC. Those are the only 2 codons that encode phenylalanine. And I'll say them again. It's TTT or TTC. Now for our degenerate primer, which is again a pool of mixed primers, we would represent our primer design in this way. I want to be clear, we now have 4 different primers that we are going to engineer into our pool. The first primer is going to have the sequence CAT-TTT. The second primer will have the sequence CAT-TTC. Third primer will be CAC-TTT. The fourth primer will be CAC-TTC. We don't know which codon encodes that first histidine, and we don't know which codon encodes that second phenylalanine. So we're making a pool of primers that has every possible combination of codon to give us a histidine followed by a phenylalanine. One of those 4 primers that I just listed a moment ago is the right primer. There's no other way to get a histidine followed by a phenylalanine. We just don't know which one of the 4 it is. We go back to our amino acid code. After that phenylalanine is a proline. Well, proline has 4 different codons that can encode it. It can be CCT, CCC, CCA, or CCG. Any one of those 4 codons will give you a proline. Now, perhaps you see where this is going already, but that means that we are now up to a total of 16 different primers in our heterogeneous pool. All 4 of the codons for proline need to be captured in every of the primer that we've already designed. So we have 16 primers. The first would be CAT-TTT-CCC. Now that encodes histidine, phenylalanine, proline. We just don't know if those are the 3 codons in our gene of interest. So we have to have the other codons in mind too, such as CAT-TTT-CCG, CAT-TTT-CCT, and down the line we go. This tree diagram shows that there are 16 possible different primers that would encode every possible combination of possible codons to give us histidine, phenylalanine, proline. I won't belabor it because I'm running out of room obviously on the screen, and I think you're getting the idea already, but we would go back and we would see that after the proline is a phenylalanine. Well, that phenylalanine we already know has 2 codons, TTT or TTC. That means that we have to branch this once again, and for each of the 16, we have to have a TTT or a TTC, giving us a total of now 32 possible primers. 32 primers, one of which anneals to our gene of interest, but we don't know which one, so we've got to make all 32. Every single primer that could possibly encode histidine, phenylalanine, proline, phenylalanine needs to be captured. Ooh, when we see a methionine, we get happy because there's only one codon for methionine, ATG. So that means for each of our 32, we just have to put an ATG after it. And then we're near the end of our sequence, and we have a lysine. Lysine can be captured or encoded by 2 different amino acids, but 2 different codons, excuse me. And they are AAA or AAG. Those 2 codons will give us a lysine. That means for each of our 32 primers, we have to also engineer an AAA or an AAG so that we encompass for every possible lysine. And that's going to give us a total of 64 primers in our heterogeneous pool. One of these primers is the right one. We don't know which one that is, but let's just play the thought experiment. That the right primer for our gene of interest was this one. In other words, the sequence of our gene on the top strand is CAT, TTC, CCA, TTC, ATG, AAG. Those 6 codons encode our protein of interest: histidine, phenylalanine, proline, phenylalanine, methionine, lysine. And that primer is the right primer. That primer anneals to our bottom strand, and it will amplify PCR. That means none of the other primers were correct. And they're all in our reaction, taking up space and taking up room. But we had no idea of that going in. We created a pool of 64 heterogeneous primers, all of which encoded histidine, phenylalanine, proline, phenylalanine, methionine, lysine. All of them encoded those 6 amino acids, but only one of them was the exact right complement of our target gene of interest. That one primer, the one that is the right complement, is the one that binds, it's the one that primes, and it's the one that can amplify our gene of interest. So the last thing to address here is the symbology, the nomenclature. Now look at this. What a mess, right? How do you order something like this? How do you represent something like this in a paper or in a way that's easy to decipher what's going on? These tree diagrams really drive home the heterogeneous pool and how every single primer that could encode that 6 amino acid chain does exist in the pool, but it's not very user-friendly or wieldy for ordering or for talking about. So we have these symbols that we use, these shorthand single-letter symbols that say, all right, this is a subset or all of the amino acids. Let's translate our primer into these symbols. Well, every single primer in our mix begins with a C and then has an A. But after that C or that A, we can either have a T or a C. We don't want any other nucleotide in there, but we can have a T or a C. We want both represented. So a T or a C, we're going to use the letter Y for that. Let me go back to our diagram. After that, we know we have 2 T's. Okay, great. And then we have another T or a C. Well, we'll put another Y in. After that, it looks like no matter which one of the 16 we're talking about, we have 2 C's. But then, after those 2 C's, we want all 4 of the possible amino acids. And that's going to be represented with an N. And then we want 2 T's in every single primer. But we want T or C represented. We know that's Y. And then we're going to follow that with an ATG no matter what, because that's the only nucleotide codon that encodes for methionine. And then we will end with 2 A's followed by an A or a G. If we want an A or a G, we need the R symbol. So this, let me full screen this for a second. This sequence here represents the entirety of these 64 different primers in a single sequence. It says for all 64, start with a C. Then for all 64, put an A. Then split the batch in half. And for half of them, put a T in the 3rd position, and the other half put a C there. Then all of those should have T's in the next 2 slots. And then in the 6th slot, split them in half again and put T's in half of them and C's in the other. Then all of them should have 2 C's. Then split the pool into even 4s, and for 1/4 of them, put an A in this position, or a G in the 2nd group, or a C, or a T. Down the line you go. So this one sequence is a much easier way to represent this heterogeneous pool than having the entirety of all of the primers in that pool represented. And interestingly enough, if we go back to our slide and we look at the primer sequence, we see the primer sequences CAY, TTY, CCN, TTY, ATG, AAR, exactly how we decoded it using those single letter codes. So that's really all we're looking for with the single letter codes. It's just letting us represent a heterogeneous pool in a nice, easy to decipher way. And really what's important is the creation of that pool, the design or engineering of that pool. So that's the idea of degenerate primer design. I hope it's a little bit clearer now than it was before, but certainly if there are still questions about it, you know where to find me, and I encourage you to ask. Thanks so much.